Hello everyone, welcome back to my YouTube channel. Today I am going to explain the lysosomes formation and autophagy. So in particular, lysosomes are formed when transport vesicle from the trans-Golgi network fuse with the late endosome, which contains molecules taken up by endocytosis at the plasma membrane. Okay. So in this picture you can see here, this is the endocytic vesicles. Early endosome, then convert into the late endosome and form the lysosome. Also, endosome play important role in intersection between the secretory pathway through the which lysosomal protein proceed first and the endocytic pathway. It is endocytic pathway directly and lysosomal formation. So, first image, you can see in this image, molecules are taken up from outside the cell here in endocytic vesicle, from the endocytic vesicles which fuse with the early endosome. Here it is early endosome. Here also you see this green color, which is the membrane receptor. So membrane receptor are recycled to the plasma membrane through the recycling endosome and early endosome. Mature, through the recycling endosomes and early endosome to mature into the late endosome. This is early endosome, the late endosome, and it is the receptor, membrane receptor, which is the recycle. Recycling receptor. Here. And they further transfer to the vesicle in vesicle form and come back to the cell membrane. Now the transport vesicle, here it is the trans-Golgi network. Right? This is transport vesicles contain the acid hydrolases enzyme. Here it is the acid hydrolases enzyme. Which fuse with the late endosome. It is and which mature into the lysosome. So lysosome are the required full component of the lysosomal enzyme. So this is how lysosome formation. It happened like this by the endocytosis process. By the endocytosis process and form the lysosome. Now second, it is the autophagy. Now in addition of degrading molecules, taken up from the outside of the cell by endocytosis. As you know, the lysosomes are responsible for autophagy. It also the turnover of the cell's own components, including cytoplasmic organelles and cytosolic protein. If you remember, I have discussed in my previous video the ubiquitin-proteasome pathway. It is the major mechanism of regulated protein degradation. So in contrast, autophagy leads to the degradation, but it is the gradual degradation of long-lived components of the cells. So in the first step, autophagy, so the attachment of the small area of the cytoplasm or cytoplasmic organelles, for example, the mitochondria. Both images are the autophagy process. So here you can see this is the mitochondrion, single mitochondria. Single is mitochondria. Here is the mitochondrion. Now the mitochondria. In this picture you can see this. This is mitochondria and covered with the endoplasmic reticulum's membrane. It is the mitochondria. Covered with the endoplasmic reticulum's double layer. This is the phagophore. And this double layer phagophore, double layer membrane and mitochondria, entire this form is the autophagosome. So here is the autophagosome. So now the autophagosomes, which is fused with the lysosome. It is the lysosome, and degrading the components of the mitochondria. It is also lysosome fused with the mitochondria and degrade the components and convert into the phagolysosome. This is the phagolysosome. And then the autolysosomes, okay, they autolyze a residual body, and then they extracells, they exit through the exocytosis process, okay, by the cell membrane. Now here approximately 1% of the cell protein per hour are degraded by this mechanism, okay, autophagy mechanism. Okay, so the damaged organelle, suppose an extra protein, okay, extra protein which is also degraded by this process. Okay. Now the function of autophagy and importance of autophagy. So the autophagy not only leads to the continuous turnover of cellular constituents, but it can also be regulated during the development and response to the stress. So for example, autophagy plays an important role in many development processes also, okay, such as insect metamorphosis, which involves the extensive tissue remodeling and degradation of cellular components. Right? This is the one function. Second function, it is in addition, autophagy is activated when cells are deprived of nutrition. Okay. Suppose in under condition of nutrient starvation, okay, so autophagy allows cell to degrade non-essential macromolecules. Okay, so their components can be reutilized by the cells. Okay, so in other circumstances, okay, so the autophagy play important role in programmed cell death. Okay, so the prevention of certain types of cancer and slowing the body aging process. Okay, so this is the main function of autophagy. Okay, now the lipofuscin granules. Okay, when it will form the lipofuscin granules within the cells. So first it appears, okay, into the lysosomes present cells first. Okay, so this lipofuscin is a type of pigment that accumulates within the cell. Okay, particularly in post-meiotic cell that do not divide. Okay, and it is a mixture. It's contained a mixture of lipids, proteins, other cellular residues. Okay, so the accumulation of lipofuscin is common feature of the aging cell and is referred to the wear and tear pigment. Okay. Now the biological function. Okay. So the biological function, it is the excessive lipofuscin accumulation has been associated with the impaired lysosomal function. Okay, in previous video also described this Gaucher disease due to the lysosomes. So first it is the accumulation by the impaired lysosomal function. Second is the observed in certain age-related disease such as neurodegenerative disease. Okay, or you can see the disorder. So this is the today's video. Okay, thank you for watching.